So good afternoon. My name is Stephen Burley, and I have the pleasure of serving as director of the Rutgers Institute for Quantitative Biomedicine and the RCSP Protein Data Bank. We're gathered here today to celebrate the 50th anniversary of the founding of the Protein Data Bank. The Protein Data Bank was established as the first open access digital data resource in all of biology, founded in 1971 with just 7 protein structures. It's become the single global archive for protein and DNA and RNA experimental structures, and today supports open access to 183,584 structures. Originally, the PDB was a US only responsibility, but in 2003, thanks to the efforts of Helen Berman, former director of the RCSP Protein Data Bank, the Worldwide Protein Data Bank Partnership was established, which enables for collaborative joint management of the PDB, EMDB, and BMRB archives by 5 different data centers around the world. 3 relating to the PDB in the US, Europe, and Japan, 1 in UK, EU, EMDB, and 1 in US and Japan, BMRB. We are fair, fact, and trust compliant and have been accredited by the Core Trust Seal. The US funded RCSP Protein Data Bank was established in 1999 using the Nucleic Acids Data Bank framework that was developed here at Rutgers by Helen Berman and the late John Westbrook. Westbrook is also credited with having developed and implemented the PDBX MMSIF data standard, which underpins all PDB activities these days. We are collaborating with the Worldwide Protein Data Bank partners in global support of more than 50,000 data depositors, and supporting millions of data consumers worldwide with our rcsp.org research-focused website. And last year, more than 800,000 educators and students with our outreach and education-focused website, pdb101.rcsp.org. We're funded by the National Science Foundation, various NIH institutes, and the Department of Energy, and hosted by Rutgers, UCSD, and UCSF. And here's a recent pre-pandemic, our most recent pre-pandemic photograph of much of the team recorded in early 2000. We've been celebrating for the last year the 50th anniversary of the Protein Data Bank, much of it with help from Rutgers and New Jersey partners. And I want to draw your attention to the fact that the Honorable Frank Pallone Jr., member of the House of Representatives for New Jersey, read in a very complimentary statement about PDB50 into the congressional record. The photograph you see here on the right with the collagen triple helix in the front is the home of the Institute and the Protein Data Bank. And you can see here Helen Berman, myself, and the then president of Rutgers, Barchi, celebrating the inauguration of this new sculpture in front of the Institute building. So it's now an honor for me to introduce my friend and mentor, Gregory A. Petsko, professor of neurology at the Romney Center for Neural Diseases in the Department of Neurology at Harvard Medical School and Brigham and Women's Hospital in Boston. Greg received his BA from Princeton University, summa cum laude in 1970, and his D.Phil from Oxford University, which he attended as a Rhodes Scholar in molecular biophysics in 1973. Prior to joining Harvard Medical School, Greg served on the faculties of Wayne State University, the Massachusetts Institute of Technology, and Weill Cornell Medical College. Greg's scientific contributions are broad-ranging. As a graduate student, he determined the first 3-dimensional structure of the enzyme triose phosphate isomerase. The PDB ID is 1 Tango India Mother, 1 TIM, laying the foundation for a 2-decade-long foray into studies of enzyme action at the atomic level, viewed through the lens of X-ray crystallography, the topic of this special lecture. More recently, his research efforts have focused on discovering new treatments for age-related neurodegenerative diseases. At last count, Greg had contributed more than 250 structures to the Protein Data Bank. His manifold contributions have been recognized with numerous awards and honors, including election to the National Academy of Sciences, the National Academy of Medicine, the American Academy of Arts and Sciences, and the American Philosophical Society. He has an honorary Doctor of Laws from Dalhousie University. He is past president of both the American Society for Biochemistry and Molecular Biology and of the International Union of Biochemistry and Molecular Biology, and past chair of the Medical Sciences section of the AAAS. Greg is a master communicator. His public lectures on aging of the population and its implications for human health have attracted a wide audience in person and on the internet. One of his TED Talks has been viewed over 1 million times. Today, he's kindly agreed to reprise his American Chemical Society PDB50 celebration lecture entitled Truth Sometimes Triumphs, A History of Structural Enzymology. Greg, welcome to Rutgers. The floor is yours.
So thank you, Steve. It's a pleasure to see so many old and not so old friends on this talk. I want to talk about the history of structural enzymology because it is so intimately intertwined with the history of the protein data bank and depends so heavily, even still to this day, on the resources of the protein data bank. And because structural enzymology was not immediately accepted by conventional biochemists or conventional physical organic chemists, I've chosen to call this lecture Truth Sometimes Triumphs, based on Max Planck's famous statement that truth never triumphs. What happens is its opponents eventually die out. That isn't actually always true. And I think this represents an interesting example of a case where it isn't quite true. So see if you agree with me after I'm done. We'll proceed now to define the concept of structural enzymology. It's a field in which structural information guides experiments aimed at how enzymes work, how they're regulated, and how they can be modulated by small molecules. The whole point is that in order to do that intelligently, you have to understand the catalytic mechanism of the enzyme. But the best route, though not the only route, and maybe even not always the best route, but usually the best route, is through an understanding of the three-dimensional structure of the enzyme, hence the name of the discipline. And as you can see, it uses a plethora of techniques, only some of which I've indicated here, to get at the question of mechanism, which in turn then informs further experiments using all of these various techniques. Now, understanding enzymes in a structural sense goes all the way back to James Sumner, who first crystallized an enzyme, Jack bean urease. The crystals from his original paper are shown there on the right. You have to understand that when Sumner did this in the 1920s, there were a large number of people who did not believe that enzymes were proteins with defined three-dimensional structures. In fact, a large number of people believed that enzymes were like colloids and were completely amorphous in a structural sense, and that this was essential for their ability to catalyze chemical reactions. After Sumner crystallized urease, he traveled to Sweden to discuss what he'd done with then the leading biochemist in the world, Svedborg, who invented the ultracentrifuge. And he walked into Svedborg's office and said, "How do you do? My name is James Sumner, and I've crystallized an enzyme." And Svedborg said, "Oh, that's remarkable. I would like to I'm hearing something in the background. Does anybody know what?
Saroj, you're unmuted. Dr. Petsko, you have to unmute yourself again.
I'm Svedborg. My name is James Sumner, and I've crystallized an enzyme. And Svedborg said that's wonderful. I'd like to talk to you about this, but just give me a moment. And Svedborg left, went out of his office, and called security because the man was obviously a dangerous lunatic. That's how Sumner's work was received. However, in 1946, he sort of got the last laugh on everybody, including Svedborg, by winning the Nobel Prize for what he'd done. But this history of structural enzymology being a kind of poor stepchild, that hasn't gone away. All right, as you'll see. From our perspective, a real understanding of how enzymes work in structural terms dates directly to the pioneering work of David Phillips, with whom I did my PhD work at Oxford University. David was the one who first solved the three-dimensional structure of an enzyme by X-ray crystallography. The enzyme, of course, was hen egg white lysozyme. And when he got the structure, they built the three-dimensional model using these Kendrew wire models. I mean, it was an exhausting experience, built into electron density maps that were sheets of glass that were the size of a human body. And he then went back to the lab that night and spent the whole night looking at the structure and thinking about how he could fit the oligosaccharide substrate into the active site of this model. And he played around with that. And during the course of that extraordinary night, he basically came up with most of the defining principles for how not only lysozyme works, but it turns out for pretty much how all enzymes work. This just shows you how things were back in the 1960s when this work was done. This is the drawing of the three-dimensional structure of the enzyme lysozyme done freehand by David's mentor, Sir Lawrence Bragg. And this drawing is actually extraordinarily accurate. We, of course, have a slightly different way of getting at this kind of information these days, but I still think this is just wonderful. These are, of course, the classic lysozyme crystals. So easy is this enzyme to crystallize that you can crystallize. I've done this by taking raw egg white and adding table salt. And if you do it right, you'll get lysozyme to crystallize out. Okay. Now here's David with his first student, Louise Johnson. And it was Louise who actually solved the structure of lysozyme with an oligosaccharide inhibitor bound. Actually, it's a product, pretty much a product of the lysozyme reaction. And this is what the structure looked like. And the whole point about this is that from this, they were able to confirm the deductions that David made by just model building the substrate into the active site with the wire models that extraordinary evening in, then, London at the Royal Institution. It allowed them to come up with a mechanism of action for the enzyme, which involved the formation of an oxocarbenium ion intermediate at the midpoint of the reaction. Now, as I said, there were multiple principles that David determined must be operating for lysozyme to effectively catalyze the hydrolysis of an oligosaccharide substrate. And these were the principles that he elucidated: proximity, general acid-base catalysis, a special microenvironment created by the enzyme for the reaction to occur, destabilization of the substrate, in which the enzymes distorted the substrate, okay, raising its energy towards that of the transition state, stabilization of the transition state, which was done electrostatically. Now, the amazing thing about that is that all of these have been shown to be general principles. Every enzyme uses them to some extent. Some enzyme uses them more than others, but they pretty much apply across the board. So we've come, and there have been a few more that have been discovered since. Nevertheless, this is an astonishing intellectual achievement. And why David never won the Nobel Prize, I have no idea. Now, what I want to do is jump about 10 years past that extraordinary achievement and go into the 1970s, when Dagmar Ringe and I started to work together on using crystallography to unravel how enzymes worked and what the specific mechanism of important classes of enzymes were. This is about 1975, 1976. Okay, and many of you weren't even born then. So I think it's very important for you to understand what life was like back then. It was a very different world, not just scientifically either. In 1976, I had long hair. Now I'm longing for hair. In 1976, I was into acid rock. Now it's acid reflux. In 1976, I was constantly thinking about going to a new hip joint. Now I may need a new hip joint. 1976, the Rolling Stones. Now, kidney stones. So it's a very different world. And for those of you who are a lot younger than I am, someday all this will be yours. At that time, in the 1970s, enzymology was dominated by a physical organic chemistry school of thought that dated back to three people: Frank Westheimer at Harvard, Bob Abels, and Bill Jenks, then at Brandeis University. And these three people, using techniques of not just biochemistry, but also, as I said, physical organic chemistry, had established the framework for how you studied enzymes. And there was no room in that framework for structure. In fact, X-ray crystallography was regarded as being uninformative for how enzymes worked. It was as though David Phillips and his extraordinary achievement didn't really exist, or if it did exist, didn't have anything to do with the kind of work that they were doing. And the arguments as to why this were true were several. I will give you the major ones here. One of the arguments was, of course, that the crystalline state was a completely unnatural environment. In fact, as far as they were concerned, it might be not even the correct fold of a protein that would choose to crystallize. And you could carry this on a bit further and say, okay, maybe it's the most common fold that crystallizes, but that might not be the fold that's catalytically active. Might be a very minor species that has nothing to do with the crystalline structure. They believed that lattice forces in the crystal would make it impossible for the protein to undergo any kind of movement that might be necessary for the chemistry. They believed that if you measured catalytic activity in a crystal, that entirely came from molecules either on the surface or more likely dissolving off the surface, and that the molecules in the crystal would have no activity. And finally, they said even if none of this was true, what difference would it make if you couldn't look at any important reaction intermediates? You couldn't look at enzyme substrate complexes, you couldn't look at transition state analog complexes, you couldn't look at anything that mattered by crystallography. Okay. So it wasn't long thereafter, and this is one reason I've chosen 76 for this part of the talk, that the first rebuttals began coming in. They came out of largely the work of Tony Fink, sadly since deceased, who was a good friend of mine and was working at the University of California, Santa Cruz, who showed that not only could you form stable crystalline enzyme substrate intermediates at low temperature, but he went on to show with Marvin Mackinnon that these intermediates actually had that these crystals actually were catalytically active. More about that in a moment. Why this matters to me is that it was at about the same time that Tony sent an undergraduate from UC Santa Cruz to work in my lab, which was then at Wayne State University in Detroit. And the undergraduate he sent was Tom Albert, who later at MIT became my first graduate student. But as an undergraduate, Tom went to the lab at Wayne State. He rode his motorcycle from Santa Cruz to Detroit in the middle of the winter, okay, to get to the university and spend a semester working in my lab on crystallography. This is what Tom looked like in 1975. Look familiar? And over the course of 6 months, Tom and Demetrius Cernoglu and I did an experiment that established that you could use crystallography to look directly at catalytic intermediates. This was the first time that had been done. We looked at elastase with a substrate bound at minus 55 degrees Celsius. This structure showed a covalent acyl enzyme at admittedly lowish resolution, bound to the active site of elastase. By the way, yes, it's true, in those days, you actually could get papers published in Nature with only 3 authors. I know that seems hard to believe today. Now, we wanted to push this to much higher resolution. But in fact, that turned out to be murderously difficult. We thought that the obvious way to push it to higher resolution was to drop the temperature even lower so we could spend much more time collecting data before this acyl enzyme deacylated, which it would still do even at minus 55. So we would drop the temperature to like minus 75, flow in substrate and collect data, and we never saw anything bound. We could repeat this experiment, but we couldn't repeat the experiment at lower temperature to get higher resolution data. It wasn't until we understood that something else was happening in the protein at around that temperature that we finally realized why that experiment couldn't be reproduced. And that's because the original experiment was done just above what turned out to be the glass transition temperature in protein dynamics that occurs right around minus 50 degrees Celsius. And all the other experiments we were doing that failed were below that temperature. And this came out of work that was done by, among other people, Ann Stock, who happens to be on this call. All right. And understanding this, and it took quite a while to understand this, made it clear how you could do the experiment so that it would work. You see, if there was a glass transition in protein dynamics, the protein was probably too rigid to even bind substrate at the lowest temperature. And that, in fact, turned out to be right. And this paper, Ann and Barney Rasmussen and Dagmar showed that this was true. But this is the point. All right. The point is, if you bound substrate at the higher temperature and then lowered the temperature below the glass transition, you could trap the enzyme substrate complex for high resolution studies. And Jiaosheng Ding, working together with Bjarne and Dagmar, were able to do this finally. And here indeed is the covalent bond between the enzyme and its substrate, now at high resolution. And this is the basis for all of the work that's been done since by people who trap intermediates in enzyme substrate complexes by flash freezing. What they're doing is they're introducing the substrate into the enzyme at a higher temperature and then flash cooling below the glass transition temperature. Below the glass transition temperature, you can't dissociate product, or for that matter, anything from the enzyme. And so this is the rationale. This is what underlies, I should say, the rationale behind all of the experiments that people now do using cryoenzymology to look at substrate complexes and intermediates. So now let's go ahead another 10 years to roughly now 1985 and look at the situation then. The situation was that despite early efforts in using crystallography to look at enzymes, that cabal of physical organic chemists who were running the mechanistic enzymology field didn't believe that crystallography had anything to say about how enzymes worked. And as you can see, there were hardly any enzyme structures in the protein data bank at that time. So what broke that stranglehold was work done by Dagmar together with Bob Abels, who was one of the members of that cabal. Dagmar, believing that you should keep your friends close, but your enemies even closer, as Don Vito Corleone mentioned in The Godfather, decided that she would rope Bob Abels into structural enzymology by getting him to see that it could do something useful for him. So working together with him, she used structural enzymology to solve a problem he couldn't solve by the classical techniques, which was how certain inhibitors of the serine proteases worked. And here's one of the first examples of that. Barbara Seaton was the student who did the work. Michael Gelb in Bob Abels' lab did the biochemistry, and the crystallography was done by Dagmar and Barbara. And it showed how the inhibitor bound to the enzyme and clarified the questions on the mechanism of action of that inhibitor. That was in 1985. And not long after that, Jim Motonen, working together with Dagmar, and again, Mike Gelb and Bob Abels did the same thing for another inhibitor. Both those papers were published in the journal Biochemistry. It had been impossible for us or any other crystallographer to get papers published in biochemistry using structural information to interpret enzymes until after these papers got in. And the reason they got in wasn't because of the crystallographers, it was because they had the blessing of Bob Abels, who was at that time the leading figure in the field. So this was a case where not only did truth triumph, but it now is, in fact, the default. Here is the cover of Biochemistry for August 24th of this year, showing, of course, the structure of an enzyme with substrate. And you can't get a paper published almost in a journal like Biochemistry nowadays without having structural information in it. In fact, I randomly looked at a few issues of Biochemistry for the last couple of years, and virtually every paper had structures. In fact, if I use the protein data bank resources, and thanks for this, Steve, you'll find that biochemistry is now the third largest contributor in terms of journals to the protein data bank. All right. So that really indicates how far the field has come since 1985. Jumping now to 2000. All right. Because 2000 is where I think the field finally came into its own, and I'm proud to say we had a lot to do with that. During the period from 1985 to 2000, our lab and other labs began to develop techniques that could be used to look at virtually any kind of enzyme substrate complex or intermediate by crystallography if you had the right conditions. And Dagmar and I, in fact, wrote a small review article about this around that time. And these were some of the most important techniques. One was direct observation of an enzyme substrate complex at equilibrium. More about that in a moment. One was establishing a pseudo steady state by flowing continuously substrate into the crystal while you were collecting data, thereby allowing whatever the lowest free energy complex was to accumulate. Another was the freeze trapping that I just talked about a moment ago, and then using techniques like Lowry diffraction or very rapid data collection with synchrotron radiation, you could actually do quasi-time resolved observation if you could trigger the reaction in the crystal, which we were able to do in some cases. This all came to a head in work by Ilma Schlichting and Ann Stock and others, which we did around 2000 to look at the three-dimensional structures at high resolution of every kinetically significant intermediate in the reaction cycle of an enzyme. And we picked the enzyme Cytochrome P450 because that enzyme had been worked on extensively by Gaston Huibon-Hoa and Pierre Duzou in Paris at low temperature. And a colleague of ours at Illinois, Steve Sligar, understood this enzyme well enough to help us figure out how to do all these things. And so over the course of a period of several years, Ilma, Ann, Joel Berenson, Kelvin Chu, with help of some others, were able to do all of the structures of the things you see here. And these are all in the protein data bank. Now, using crystallography in that way was somewhat new. There was still an effort involved in solving protein structures at that time. It wasn't as easy as it's become. And so choosing to do something like this wasn't a trivial choice, but it was made much easier by the existence of the protein data bank, because we were able to exploit the fact that the structure of P450 had already been solved in several different crystal forms. And although none of those crystal forms were suitable for this kind of procedure, ILMA was able to get a new crystal form which could rapidly be solved by using the existing three-dimensional structure. And here, just to show you, are all the electron density maps of all the different complexes that were done. This is the structure we used to solve all those structures. This was Tom Poulis's structure of the camphor P450 with, in fact, the heme bound to a water molecule. And that was sufficient to get us started, but it also saved us an enormous amount of time because we could solve all these other different crystal forms by just using molecular replacement. And now, taken from the protein data bank, here are some of the structures. Here is the P450 enzyme in its resting state with nothing bound to the iron in the reduced form. Here is what the first binding of oxygen, molecular oxygen. Okay. This is the change that occurs in the enzyme when oxygen binds. It pushes the substrate away, all right, and moves the iron into the plane of the heme. And all of these things you could see at high enough resolution. And two new water molecules appeared in the structure. There was room for them when these motions occurred. That was exciting because it was known that this enzyme reaction required protons, but nobody knew where the protons came from. Well, it turns out they came from the water molecules. And that's how they were positioned properly in order to do the chemistry. And all of these things you could learn by looking at the structure of the intermediates at high resolution. It was really very exciting. And we even got the structure of the high energy oxoferal intermediate. People knew this intermediate existed, but there were a lot of arguments about what it looked like. Here's the structure, the difference map between that structure and the dioxygen intermediate. You could see one of the oxygen atoms has been lost, as it has to be when this bond is cleaved. And the end result is now this, an oxime intermediate. We knew all of its parameters. It's really quite wonderful. And it's positioned perfectly to drive this oxygen atom into the carbon-hydrogen bond of camphor, hydroxylating that molecule, even though this bond has not been activated in any way. All right, this high energy oxygen can do the job on its own. So we were really quite delighted with this result. And for those of you who want to read more about it, here's the paper, okay, in Science, published in 2000. These are the people involved. I should definitely mention Bob Sweet, who was absolutely essential for getting this work done at the Brookhaven synchrotron, which beamline he managed at that time. Now, at the same time, we were using other techniques, and so were other people, to look at other intermediates and to look at other aspects of enzyme mechanisms. We did a lot of work on triose-phosphate isomerase with Jeremy Knowles at Harvard. And we could do that because that's a single substrate, single product reaction. You didn't need any special techniques. You just flowed substrate into the crystal. And whatever the lowest free energy species was, it would accumulate. And that's just an equilibrium between these two molecules. This is the mechanism that was worked out for triose-phosphate isomerase. Why I'm mentioning this is that at exactly this time, in the mid-1980s, people began using the then new technique of site-directed mutagenesis to test ideas about this mechanism. For example, one point of this mechanism was that there was a catalytic base, a glutamic acid, that abstracted a proton from the substrate. So Jeremy mutated that glutamate to aspartic acid, moving the catalytic base backward by 0.7 of an angstrom. That's all he did. And we could show by crystallography that it didn't do anything else. Well, moving the catalytic base back by 7/10 of an angstrom dropped the catalytic rate by over 1,000-fold, showing the importance of proximity and orientation for the reaction of an enzyme like this. Subsequently, people like Dan Hirschlag, who happened to have been a student of Bill Jenks, one of the original cabal of physical organic chemists, have used techniques like this to probe in much more detail critical factors like how short and strong are the hydrogen bonds to transition state-like species in the active sites of enzymes. And so you're beginning to get a level of fine probing of the mechanistic features that an enzyme uses that would have been unimaginable in the 1960s when mechanistic enzymology began. So if we look at what's happened since lysozyme, with all of these techniques being used, here's what we know. We know that all enzymes use proximity and orientation. That is, in fact, the universal characteristic. They hold things close enough together for reactions to occur in the right way. We know that most enzymes use general acid-base catalysis. We know that all enzymes create a unique microenvironment apart from the bulk solvent where these reactions would occur without an enzyme. We know that many enzymes destabilize their substrates by straining them in a conformation closer to that of a reaction intermediate. We know that most enzymes stabilize the transition state, usually electrostatically. And then something we've learned more recently is that many enzymes make heavy use of dynamic properties of proteins to do a lot of things connected with catalysis. And there's where the protein data bank becomes interesting. Again, because if dynamics is important for an enzyme, then it may not be enough to just have one structure in the protein data bank. If the enzyme is able to sample a number of different conformations, all of those conformations could be important. And so it may not be the person who solves the first structure of a protein who solves the right structure or the most important structure of that protein. That may turn out to be something quite different. Let me give you an interesting example of that. This is the drug Gleevec, which is the first personalized medicine drug. It is a drug used for chronic myelogenous leukemia and also for gastrointestinal stromal cancer. It was developed by Novartis, and it is an inhibitor of the BRCA-ABL tyrosine kinase. Well, it turns out Gleevec was found by screening. You could not have designed Gleevec to fit into the active site of the kinase that it inhibits, because at that time in the protein data bank, there was a structure for that kinase, but it turns out, as John Currian and his associates shown, those kinases are quite flexible, and they adopt more than one conformation. Only one conformation was in the protein data bank at the time, and it turns out that Gleevec binds to the other conformation, which hadn't been seen until it was trapped by the drug. So no matter what's in the data bank now for an enzyme, it is quite possible that there is room for more things to be done with that enzyme and for additional structural work to shed light on, if not the workings of the enzyme itself, how it might be exploited pharmacologically. Therefore, the database is going to grow, not just to add new structures, but to add new states of existing structures. This is important. Now in 2021, I want to talk about where we're going. All right. I've told you where we're at. I want to talk about where structural enzymology is headed. One of the most important things I think we need to worry about is the fact that nearly all of the structures that have been done since the 1980s have been done under cryo conditions, not just to trap intermediates, but in many cases, most cases, in fact, the structure of the parent enzyme itself with nothing bound. And that's because, of course, with synchrotron radiation, most people use cryo conditions so that you can get data sets from single crystals without radiation damage destroying the crystal. But the worry has become, for some of us, that in so doing, we may be either reducing or in some cases eliminating essential aspects of the dynamics of these proteins that would occur at physiological temperatures and be important for catalysis. Almost going back to the original argument of the physical organic chemists that maybe things are a little too rigid, but not because of the crystalline state. Rather, they're too rigid because of the temperature. And in one of the last papers that he wrote before he died tragically of ALS, my first student, Tom Albert, then professor at Berkeley, together with his student, Jim Fraser, and others, looked at this question and came to the conclusion that we might well be missing things that were interesting and important by never looking at protein structures at more nearly physiological temperatures. All right, here's some of the conclusions from that paper. We found that crystal cryocooling remodels the conformational distributions of more than 35% of side chains and eliminates packing defects, that is to say, cavities in the protein that may be necessary for functional motions. In the signaling switch protein RAS, an allosteric network, has been uncovered. By the way, there's some very exciting work on that done by Carla Matos, who's on this call. But the point is, this sort of thing, this reliance on low temperatures, expose a bias in structural databases towards smaller, because the protein shrinks with temperature, overpacked, and unrealistically unique models. Here's an example from some work by Mark Wilson, together with Jim Fraser, on the enzyme E. coli dihydrofolate reductase. They went to the trouble to determine the structure not at cryo temperatures, but at cold room temperature. And what you can see are an enormous richness of motions. This was done at close to 1 angstrom resolution, so you can fit thermal ellipsoids to all of the atoms in the structure. These are much bigger than you'd ever see at cryo temperatures, where you just get tiny little spheres for most of your thermal ellipsoids. And the fascinating thing about them is that many of these ellipsoids, this one, for example, are along the reaction coordinate. They're oriented in such a way that the motions will take the atoms where they need to go for the chemistry to occur. These are the sorts of things we may be not seeing and not understanding as well as we might by over-relying on cryo conditions. So my conclusion is, on that, we should take more advantage of high protein concentration in protein crystals. You know, the packing of molecules in a crystal is very similar to the packing of proteins in a living cell. And we should be exploiting that more. Protein crystals are great reaction vessels to do high concentration enzymology. We should study the properties of proteins in crowded environments like crystals. But more to the point, we should also study enzymes inside living cells, and we should do crystallography at more nearly physiological temperatures. We have the techniques to do that now. I think if we did, we'd be seeing some extraordinary things. Look, this is a drawing of how crowded things are inside a living cell. And here's the packing in a protein crystal. If anything, it's less crowded than it is inside a living cell. In fact, the density of molecules in a living cell approximately the same as that of the density of people in Times Square on New Year's Eve. We should be thinking about this more. And this plays a role because I think what's going to happen, where the future is going to take us, is we're going to be trying to do structural biology and structural enzymology in vivo, or in conditions that more nearly approximate in vivo conditions. Here, from Science last year, is a paper using cryo-electron tomography to look at a multi-protein complex inside a living cell. Okay. And it's quite extraordinary. Here are the particles, and here are the reconstructions. And what this is a structure that couples transcription and translation. Been known for a long time that transcription and translation were coupled in a biological sense inside bacteria. What this paper showed is that this coupling was also structural, that the ribosome was bound to RNA polymerase, and that you would see the transcriptional complex and the translational complex physically associate with the help of some other proteins. Remarkable. Okay, and I think this is where we have to begin to go with structural enzymology. So with that in mind, and with a deep thanks to all of the people who've worked with me over the years, some of whom are on this call, some of whom I've mentioned, and many of whom I didn't have time to mention, I'm sorry about that, but whose work has been absolutely essential. Let me wish the Protein Data Bank a happy 50th birthday. I remember when I was 50. And now I realize that was still pretty young. So I think there's a good chance that the Protein Data Bank is also still pretty young, and that it has a long way, I hope, to go. In another 50 years, let's do this again. Thank you very much.
Thank you, Greg. So let me, to be respectful of everybody's time, I need to draw this to a close, and I want to just take a moment to provide some acknowledgements and thanks. First, Greg, thank you very much for a scintillating reprise of your ACA meeting PDB50 celebration talk. We had more than 140 unique participants joining the session. Thank you all. And I have, in terms of logistics, to thank Christine Sardecki, Maria Voigt, Shamara Whetstone, Michelle Sangera, and Steve Odinsky. I'll reiterate my thanks to our funders at NSF, the NIH, DOE, and more recently, the Rutgers Cancer Institute of New Jersey, our hosts at the Rutgers Institute for Quantitative Biomedicine, UCSD and UCSF, and the PDB50 sponsors shown on the next page. You can continue the buzz if you would like. We have 2 more PDB50 events scheduled before the end of the calendar year, November 16 and 18, working with the Royal Society of Chemistry and Protein Data Bank in Europe. We're putting on a 2-session training opportunity for students focused on the topics listed here. And then on the 24th of November at about midnight East Coast time, our colleagues at Protein Data Bank Japan, based at Osaka University, will be hosting an event at the Biophysical Society of Japan. And you can find out more about these events at rcsb.org/PDB50. We have 1 more crash course to go for this year, and it, not surprisingly, is focused on protein structure prediction. And there'll be both didactic lectures, including one from the Rosetta Fold author at the Baker Lab, and an opportunity for doing hands-on protein structure prediction with both AlphaFold and Rosetta Fold using the Rutgers Amaral cluster, if you have a Rutgers ID. I need to acknowledge the fact that there are a very large number of people that allow me to stand at the tip of the spear here at RCSB Protein Data Bank. I want to thank all of these talented individuals and reflect again on our sad loss of John Westbrook on the 18th of October, who died suddenly from a heart attack from which he could not be resuscitated. He will be sorely missed. Those of you who are interested in considering employment opportunities with the RCSB Protein Data Bank, please go to our jobs page, www.rcsb.org/pages/jobs. We've got openings for a cloud systems administrator here at Rutgers, scientific software developers at UCSD, and a postdoctoral fellow skilled in computational structural biology at UCSD. And with that, I will thank you all for listening. And thank you again, Greg, for a wonderful tour through nearly 50 years of structural biology. Be safe.